Hey guys, welcome to the video on multiple alleles. So just as a reminder, we're going to be doing something that is similar to a monohybrid cross, okay? But more focused like the incodominant and incomplete dominant and codominant, where it's going to have a set letter and each of those letters will have a changing allele attached to it. So let's just jump in. So what are essentially multiple alleles? So multiple alleles is a gene that has more than two alleles attached to it. This is actually more true in our body and is actually more how inheritance happens than anything else. In fact, it was actually very lucky that Mendel, the father of genetics, actually figured out that peas using plants was what caused you to do crosses, because normally multiple alleles is what is used. So we have a ton of examples. We're going to stick to a few that are simple to see and the ones that are most commonly used. So that is the eye color in Drosophila, which are fruit flies, which if you, again, move from here to university and take a genetics course here, you're going to love hanging out with fruit flies. Hopefully you understood the sarcasm. Fruit flies suck. Now, the next one is blood types in humans. So you may remember blood types from Bio 20, and we talk about A, B, and O, and how there was both R negative and RH negative and RH plus. Well, they're considered multiple alleles because although A and B are in a codominant relationship, when you have them together, they're both expressed. A and B are actually dominant to O. That relationship is not multiple allele, but what is multiple allele is when you have your blood type plus your RH factor. So that's actually a type of multiple allele. So we're going to look at the one that is used most commonly, which is eye color in Drosophila. For quizzes and tests, I will give you kind of a key for this simply because it is not easy to remember. But you also have to remember that eye color and Drosophila are going to have a dominant recessive relationship as well as being a multiple allele. So what does that mean? Well, if you look on the side there, I have included a legend. The legend is showing you all the different eye colors that you have in fruit fly. So here is my wild type, which is my red. This is apricot here. No, this one's apricot, honey, and then this is white. Okay. And you'll notice that they're labeled 1, 2, 3, 4. The reason they're labeled 1, 2, 3, 4 is because one is dominant to 2, and one is dominant to 3, and one is dominant to 4. And it moves down the line like that. So 2 is dominant to 3, and 3 is dominant to 4. So how do we know what are the possible phenotypes genotypes of fruit flies? So there's all the phenotypes listed. So if I have a fruit fly with red eyes, or what we call wild type eyes, these are all the combinations of genes I can have. I can have 2 dominant E1s, which we would call purebred or pure breeding, because they're both homozygous. They're the exact same allele. I can have what we would call a heterozygous individual, right? So E1, E2, E1, E3, and E1, E4. All right. All because of this has E1 in it. We know that it's going to be wild because E1 is dominant to all the other versions. Whereas apricot, you will notice, has one less combination. And that is simply because if I have E1, it's just wild type. So for apricot, any fruit fly that has E2 in it, as long as E2 is bigger than 3 or 4, it's going to express apricot eyes. This repeats itself all the way down the pattern. So honey is either E3, E3 or E3, E4, and white is either only E4 because it is recessive. So make sure you put these down into your notes, as we will, again, sometimes I will give you a little bit of a cheat sheet for this, but for the most part, if you can remember the legend on the side, you should be fine. So we're going to go through an example of how to do a multiple allele cross. So what it asks you to do is predict the phenotypic outcome if a honey-colored heterozygous fly is crossed with a white fly. So what are we going to do? I don't know. Let's go back and look. So if we go back to our legend in the last one, we're looking here, okay? White is E4s. So if I look at my genotypes, my only option is E4, E4. And the other one said wild type. So it says predict the phenotypic outcome if a honey-colored heterozygous fly is crossed with a white fly. So I'm just going to go back one more time. Here is honey. Honey, if we look here, it refers to it as the heterozygous version. So we have to choose E3, E4 because this is the purebred version or pure breeding. It has the same allele twice. Sorry for flipping back and forth so much. So what do? Well, you know the rules. Write out your legend, put it along the side. Next, let's make the genotypes of the parents. So I said that a heterozygous fly would be E3, E4, and a white fly is the recessive version, so just E4, E4. Let's make our gametes. So E3, E4, and 2 E4s. Let's plug those gametes into our Punnett square. So there's my Punnett square. Here is my first possible offspring, my second, my third, and my fourth. All right. So we are predicting the phenotypic outcome. Phenotypic is what we see. So if we look at this chart right here, in this column here, I can see E3 comes before E4. Therefore, it will express the honey color eye. So honey color here, and I will notice both of these are E4s. So this is white. So we have a 1-to-1 ratio of honey to white. Take a moment to pause the video. Make sure this example is in your notes 100%. And there's my phenotypic ratio. All right, example 2. What are the possible phenotypes of parents in a cross which produce the following offspring? So I would maybe pause the video here, try this problem on your own. If you're thinking, I have no clue, then just follow along and fill out your notes. So let's look at the Punnett square. You will notice that this Punnett square has E1s, E3s, E2s, and all these lovely, okay? So how can we predict the parental outcome? Well, it's pretty easy. Look at the very first allele in every box. So here's this option. Here's this option. You'll notice E1 is the same in both of these. So my prediction is that E1 was one of the gametes over here on the side. I'm then going to go down here and say E2, E3. They don't have anything in common, so I'm not going to guess just yet. Okay? I'm going to come back up here, though, and notice that there's an E2 in this box and an E2 in this box. I know that this allele here, up here, is probably going to be E2. And the reason it's written second here is because it's recessive to E1. And the reason it's written first here is because it's dominant to E3. So right now I've predicted E1 over here, E2 over here. So that's what it looks like so far. I'm then going to go back. I now know I have to have a couple other options. The rest of my options look like 3s. So I'm going to guess E3, E3. So let's see if my predictions are correct. Perfect. They are. I know what you're thinking. She made this question, so of course she knew what the answer was. All right. I did, but I explained my thinking out loud to you. So remember, try to, when you're walking through genetics questions, to think to yourself, how do I solve this? Start by looking for commonalities. All right. So parents would be, in this case, wild, which is E1, E3, and apricot, which is E2, E3. Again, I'm just using my legend from the very first example that we did. So blood typing in humans is a codominant relationship, which you learned about the other day. But what you have to remember is it's another example of multiple alleles because it's actually controlled by 3 different alleles. Okay, so AB blood and I are in a recessive dominant relationship. A and B are co-dominant. They kind of work together. And that is shown when you have type AB blood. And like I already said, O is recessive to A and B. So this is what it looks like. When you have multiple alleles for blood types, Okay, and to be honest, guys, blood typing, we keep it simple in Bio30. There's actually lots of different ways to look at blood types. We just keep it the simplest way possible, okay? There's actually different versions of blood typing that are part of your actual blood, but we keep it with what is your blood type. So when we look at this multiple allele relationship, you'll notice that these are very similar, and that is because they were in a co-dominant relationship. Neither of them are better than each other. Okay, so they have the same outcomes of dominant recessive. A's are either purebred A's or heterozygous with the O. B's are purebred B's with a heterozygous with an O over here. Then we have the co-dominant relationship. So there isn't another one over here because they're just co-dominant to each other. And then I, two little i's makes the blood type O, is the recessive form of everything. So let's look at how we could predict an outcome of the person of blood type AB that mates with a person with the blood type O. So again, let's make our parents. All right, so we know blood type AB, we know blood type O. Let's make our gametes, plug them into our Punnett square. Oh, a little too fast on the trigger here. So what you'll notice is as we cross, we have type A, type B, type A, and type B. So in this example, we can only have a phenotypic outcome of 1-to-1, type A to type B. So again, this is what I see. Their genetic or their genotypic ratio would be the same, 1-to-1. So what we're going to do is find the genotypes and phenotypes of everybody involved in this question. So mom is blood type A, dad is unknown, baby is blood type B. Mom is blood type B, dad is unknown, baby is blood type O. So start by writing down what you know for multiple alleles. Mom could be either or. She's either purebred A or she's a heterozygous A individual. We don't know at this point, so write both down. Dad is unknown, but we do have some information about baby. We do know baby is either going to be big B, so purebred for blood type B, or is going to show the heterozygous form. Looking at this, I can tell you right now that baby got a little I from mom, and therefore mom has to be the heterozygous form because it got that little I right here, which of course is covered by that, right from mom, because you'll notice that it's blood type B. Mom doesn't have a B to offer, so that means dad has to be one of these two options. I should say three options, sorry. So dad has to be either big, purebred for B blood, heterozygous for B, or it could be AB. We are not given a full enough amount of information to solve this, but we know dad has to have some sort of B blood. Okay, moving on to blood type of the other side. So again, write out your types of blood types. Which one could mom be? Let's look at baby. Baby has to receive two little letter O's. Okay, now this is where it's really tough because we know in order to get the recessive type, dad also has to have a O. So that means dad could be blood type B, that's heterozygous, blood type A, heterozygous, or just O. Again, we don't know for sure, but we do know conclusively which ones both the mums are. So hopefully these examples made sense, all the multiple alleles. You're going to work through your practice questions and we are going to mark them the following day. I hope you guys have a fantastic evening.